Hello and welcome to the video looking at what effects temperature has on the membrane permeability and also a bit about how the temperature can affect the structure of the membrane. So just to mention that temperature, if we're increasing the temperature, we're giving more kinetic energy to particles and they're going to move faster. So decreasing the temperature lowers the kinetic energy of the molecules, causing them to move more slowly. And that's a basis that we're going to base a lot of this video on. So when we're looking at how, I'm going to do temperature decreasing first because it's slightly different. So when the temperature decreases, saturated fatty acids become compressed. And the unsaturated fatty acids, making up the cell membrane phospholipid bilayer, become compressed, and the kinks in their tails push adjacent phospholipid molecules away, and that maintains the fluidity of the membrane. So the kinks are in the tails and basically just maintain the fluidity. So we can see how the proportions of saturated to unsaturated fatty acids in the bilayer will determine what the membrane's permeability and fluidity would be like at cold temperatures. So it's very much dependent on these proportions. And then we can also see that cholesterol is a buffer, so it buffers the effect of lower temperature to prevent a reduction in the membrane's fluidity. So you can see how the components really do affect how an organism would deal in temperature drops. So some organisms, such as fish, can change the composition of their fatty acids in their cell membranes in response to lower temperatures, whereas some organisms cannot. If we're looking at temperature increase, then this is more straightforward. The phospholipids acquire more kinetic energy and they move around more. So they move more, and as they're moving more in a random way, they're increasing the membrane fluidity and the permeability increases. It also affects the way the membrane embedded proteins are positioned and may function, but we'll have a look at that in a second. So an increase in membrane fluidity might affect the unfolding of the plasma membrane during phagocytosis, so might affect unfolding of might affect phagocytosis, so for example endocytosis, the bending of the membrane around like a bacterium. An increase in membrane fluidity may also change the ability of cells to signal to other cells by releasing chemicals. So again, we've got exocytosis of releasing chemicals, cell signalling are all going to be affected by this temperature increase. Because if we're looking at proteins, I'm going to change to a different colour here. Proteins and the temperature, we can see that if the temperature was to increase, and they're going to have more kinetic energy, they're going to be vibrating and moving a lot more. This strong vibrations might actually break the hydrogen bonds, and the ionic bonds as well in the tertiary structure of this protein. And as we know from the enzymes topic, they can unfold, and the overall function and shape of if the shape of the protein changes, then the function of the protein will also change, and the protein might become denatured if it's an enzyme, for example. So we can see that if it was a channel protein, for example, through the plasma membrane, if that structure changed, it might let more things through than it should. And we can see this particularly through one experiment that looks at beetroot. I'll just go through that here quickly for you in this space. Right, so we've got the beetroot cells. We can see that they've obviously got dye, haven't they? I'm going to put it in purple. So if you cut the beetroot into similar sizes and you soak them in beakers of distilled water, remembering that distilled water has the highest water potential, if you had beetroot in here, you would set up a series of water baths at different temperatures, and collect 3 or 4 beetroot cores from the beaker, and we would put them in different temperatures. So we might have one at 10 degrees, one at 20, one at 30, 40, etc. And then after a set amount of time, so you might do it overnight, you might do it for 1 hour, it depends. You'd remove the beetroot and you would block them with a paper towel, and then you would use a colorimeter. So you would now have in your test tube, you might have a solution that looks a bit more purpley, because the actual kind of fluid that has been let through the membrane is now gone into the solution. So we use a colorimeter to and you'd set that to a blue or green filter, but these will be in your practical skills. And then you would check the colorimeter reading for distilled water and measure the absorption. So the absorption of each test tube, and plot a graph of absorption against temperature. So if there was less light that could pass through the solution, then we know that the solution is more pigmented, and if it's more pigmented, then there has been more colour and pigment that's leaked out of the membrane. So that's how we're showing So if the absorption was the lowest at 30 degrees, then we know that that's probably when the structures will start to denature and become the most permeable. Thank you for watching.